Now we will come to the eukaryotic cells or the eukaryotic organisms. Organisms made up of eukaryotic cells are called eukaryotic organisms. Karyon means cell, as we know. Eu means new or latest. So the new cell or the latest cell in terms of evolution are known as eukaryotic cells. Now, eukaryotic organisms can be unicellular, starting from a blue-green algae, starting from a protozoa to humans to a blue whale to a big ficus plant that we can see. All are eukaryotic. So eukaryotic organisms can be both unicellular and multicellular as well. Now coming back to the features of the eukaryotic cell, they have membrane bound organelles. They have a cell wall. All the plants coming under the eukaryotes, all the plants coming under eukaryotes, they have a cell wall made up of cellulose. So we can say a cellulosic cell wall. Students, you also have to keep in mind of another point. Fungus or the kingdom fungi, they also have a cell wall and they also are eukaryotes, but they do not have cellulose. They have peptidoglycans, they have mucopolypeptides. So that is also a cell wall, a eukaryotic cell wall that is not made up of cellulose. So here we can say except fungi, since they have a cellulosic cell wall. Now, we know human cells have a nucleus. All eukaryotic cells have a well-defined nucleus. Well-defined nucleus. Also, eukaryotic cells have 80S ribosomes, meaning they have two subunits. The 80S ribosome has two subunits. The larger subunit is 60S, the smaller subunit is 40S. Eukaryotic cells can be kingdom Protista, kingdom fungi, kingdom plantae, kingdom animalia. But in this chapter, we don't need to look into the fungal cells. We don't need to look into the protist cells. So we can omit those two kingdoms and we can focus on plant cells and animal cells. So eukaryotic cells does include two other kingdoms, kingdom Protista, kingdom fungi. But with respect to this chapter, we need to study plant cells and animal cells only. So, what is a plant cell? What is an animal cell? To be very precise, all the plants that you see, starting from the mosses, starting from the ferns, to the plants we have in our gardens, the ornamental plants such as rose, such as hibiscus, such as gulmohar, such as various orchids, to huge trees like pines, cedars, deodars, the ficus trees that make up our forests, to the plants that provide us food and vegetables and fruits like rice, wheat, tomato, brinjal, coconut, mango. Every plant that you see and which you probably have not seen in your life, everything is made up of plant cells. Now, plant cells have a few unique features that separates them from animal cells. What are those? Let's look into them. Firstly, a plant cell has a cellulosic, a cellulosic cell wall. Animal cells lack that. Animal cells only have a cell membrane. Plant cells also have a cell membrane. If you look into a plant cell, you will find a huge central vacuole. If this is a plant cell, this is the cell wall. Here we can find a huge vacuole. This is a vacuole or a central vacuole. This thing is absent in animals. Animal cells have small, tiny, this is the nucleus, this is the nucleus. They have small, tiny vacuoles that are temporary. They are termed as food vacuoles or phagocytic or pinocytic vacuoles. Now, what is phagocytosis and what is pinocytosis? If a cell engulfs a solid food material, it is termed as cell eating or phagocytosis. Like when we eat something solid, we term as eating. But when we drink a liquid, water, coke, juice, anything, it is referred to as drinking. Similarly, when a cell engulfs a liquid material, it is termed as pinocytosis. Now, the food, the water, everything a cell ingests, they store it in temporary vacuoles until and unless it is processed or digested. After that, these vacuoles are obliterated. They cease to exist. So, animal cells have temporary food vacuoles or phagocytic vacuoles or pinocytic vacuoles. Plant cells have chloroplasts. Chloroplasts, which is absent here. Chloroplasts contain chlorophyll, which helps a plant to make its own food through photosynthesis. That is why plants are known as autotrophs. But since chloroplasts and subsequently chlorophyll are absent in animal cells, they are termed as heterotrophs. Animals like us, we can't make our own food. Also, there are a few things that are solely present in an animal cell but absent in plant cell. What are those? Animal cells have centromeres. Plant cells do not have centromeres. Animal cells also contain a different type of protein processing unit. Here it is known as Golgi apparatus, here it is known as victiosomes. So these are the few basic things that define a plant and an animal. Also, animal cells and plant cells have different sets of enzymes. So chemically they are also different, but we will come into those parts when we will discuss each organelle in detail. Okay, so now let's look into the inside of a cell. How a cell looks from the inside. First, when we will observe a plant cell, let's draw a plant cell first. we will see in a plant cell that there is a cell wall. Cell wall is made up of cellulose, as we know, and also contains a deposits of other materials that solidifies the cell wall, such as lignin, peptin, suberin, etc. The cell wall primarily gives shape to the cell and gives it mechanical strength and support. Now, coming inside a cell, we will also find a cell membrane. This region of a cell is known as the cell membrane. Now, a plant cell wall also has a structure that helps it to connect with other cells. It has got tiny pores or tiny gaps in certain parts that helps it to communicate with other cells. These gaps are known as plasmodesmata. Plasmodesmata. Now coming to the cell membrane. Cell membrane is the outermost covering of the animal cells. So we will look into the cell membrane from the perspective of an animal cell. Let's see. Here we are drawing an animal cell. Animal cells, as we know, lack a cell wall. So the only thing an animal cell has is the cell membrane. In plants, the cell wall was discovered by Robert Hooke. But in animal cells, the cell membrane has a specific model known as the fluid mosaic model, which was given by Singer and Nicholson. We will look into that model very shortly. So this is the cell membrane or the plasma membrane. The plasma membrane, if we can magnify it, looks as a layer. Like we have a cake, if we cut the cake, we can see the different layers. If we cut a cell in a transverse section, we can see the layers of the plasma membrane. How does it look? As proposed by Singer and Nicholson, the cell membrane is made up of a lipid bilayer. A lipid bilayer. As we know, a lipid has a head and a tail. These heads are hydrophilic. So they face outwards and inwards of a cell, which is mostly filled with fluids. But the tail regions are hydrophobic. So they face inwards towards each other to avoid water. Also, there are proteins spanning throughout the cell membrane or on top of the cell membrane. This is known as intrinsic protein. This is known as extrinsic protein. Intrinsic proteins are also known as transmembrane proteins, and these are known as surface proteins. There are also other proteins that form channels inside a cell. They are known as receptor or channel proteins. Now, these proteins are also associated with carbohydrate chains, mainly made up of sugar. So, these proteins are also known as glycoproteins. Glyco meaning carbohydrate chain and protein means protein. So, this is a protein, lipid, and again protein bilayer. This is known as the fluid mosaic model of a cell membrane or plasma membrane given by Singer and Nicholson.